Okay, so we're going to need to get through some important information talking about cancer. And we're going to start off with talking about the cell cycle that we just talked about, regulation of mitosis, right? Mitosis has to be triggered, and once you trigger a cell to go through mitosis, you have to stop. You have to remove the trigger. So you effectively have to turn on and turn off mitosis. So in turning on mitosis, you release something creatively called a start signal. So that activates mitosis. So when you turn on mitosis, you send a start signal, and that causes cells in the area to start undergoing mitosis. Stimulates mitosis. Cells cannot start mitosis on their own. They require a start signal. If you get a cut and you need more skin cells to fill in the wound, they will not automatically start mitosis. The cells that undergo mitosis need the start signal, and then they can start producing cells that will become skin cells that will fill in the wound. So, turning off mitosis also requires signals. Who wants to guess at the signal to turn off mitosis? It's not a start signal, it's a
stop
signal. There you go. Stop signals. Stop signals turn off mitosis. They deactivate mitosis. And again, once you send a start signal out there, mitosis is going to happen, and a stop signal is required to stop mitosis. There is something called density-dependent inhibition, basically where your cells, once they get close enough, there's little stop signals on the membranes. So when they get close enough to contact each other, the start signals are right there on the membranes, and that turns it off. So that's density-dependent. Basically, when you get them smooshed up against each other, it turns off. They're still stop signals, but they're on the membranes. And so as long as they're not in contact with each other, mitosis occurs. But once they get crowded enough to get in contact with each other, they get the stop signals. My battery is running low. That is not a good thing. Aha! The cord is disconnected. Give me one moment to free myself. There we go. All right. So we require start signals and we require stop signals. Whether the stop signals are released upon the cells or the stop signals are on the membrane and smooshing against each other when they're crowded enough causes the stop signals. Start signals, stop signals. Another important thing to talk about is how mitosis occurs in a human body, right? So mitosis occurs in specific cells. Specific cells can do mitosis. So not all cells can undergo mitosis. The skin cells in your, on your arm, all those skin cells, they cannot undergo mitosis on their own. So only specific cells can do mitosis. This results in what we call differentiation. Process of cells becoming different, amazingly enough. So you start with a progenitor cell, a stem cell. A stem cell can do infinite mitosis. Send a start signal to a stem cell, it cranks out a new cell. It cranks out cells, right? So, thing about stem cell mitosis pathways is that they end up producing another cell with a limited number of mitosis events. So this is the daughter cell. Stem cells infinite, daughter cells finite. Daughter cells produce a differentiated cell. Think of it like the final form. And it cannot do mitosis. So if you think about it, there's a really easy example that would be blood. So in your bone marrow, inside your bone marrow, there's two different types of bone marrow. There's yellow marrow, which is just like fatty storage. And then there's red marrow. Red bone marrow is where you produce blood cells. So you have a blood stem cell. Say you've lost blood and you need new red blood cells, right? So the blood stem cell in bone marrow divides. And you get some daughter cells. And that one divides. And usually what happens is you have, 1 to 2 more divisions before you end up with a red blood cell. Right? So this stem cell in the blood, in the bone marrow, the blood stem cell, it can become it has a differentiation pathway that produces red blood cells. But it also produces all of the white blood cells. Macrophages, neutrophils, granulocytes, that's lots of stuff you don't necessarily need to know. But it produces red blood cells, it produces the white blood cells, and it ends up producing platelets as well. So its differentiation pathway takes it to quite a few different potential cells. Do I have that diagram? I don't. I should potentially get that put in here. Let me see if I can pop it up on the internet. Let's see. Blood cell differentiation. Images. There we go. That looks good. Open image in new tab. Wow, that looks terrible. They had a white background on that one, huh? There we go. That one will work. No, it is tiny. Wow, this is taking much more time than I thought it would. Okay, last chance. Okay, there we go. Let me change what I am sharing. And there we go. So this HSC right here is called a hemopythetic stem cell. Yeah, complicated name, I know. And so it can produce this daughter cell. And this daughter cell can produce an L blast, which can become a lymphocyte, a white blood cell, or this progenitor cell here. And that can become a myeloblast, an MO blast, or a pro-M blast, getting you a monocyte, a type of white blood cell, a neutrophil, a type of white blood cell, or eosinophils and basophils, which are types of white blood cells. So this differentiation pathway gets you 4 different kinds of white blood cells, all from the hematopoietic stem cell. Now, if you move past this second daughter cell to another daughter cell, you can get promegakaryocytes, which is a progenitor for the megakaryocyte, which is a massive cell that's fate is to break apart into itty bits. And those itty bits are the actual platelets. And if you want to make a red blood cell, this progenitor becomes a pro-E, baso-E, poly-E, ortho-E, and then finally a red blood cell, an erythrocyte. So you can see it's a complicated pathway. So when you send a start signal out to get yourself white blood cells, red blood cells, or platelets, that start signal affects a stem cell. Red blood cells don't undergo mitosis. Megakaryocytes don't undergo mitosis. Eosinophils, neutrophils, monocytes, lymphocytes, they don't undergo mitosis, right? To get more of any of those cells, you gotta start at the undifferentiated stem cell. All right, let's get back to where we were, the differentiation pathway. A stem cell can undergo infinite mitosis, is considered undifferentiated, because its mitosis can get us lots of different cells. So the differentiation pathway is how we go from an undifferentiated cell to a differentiated cell. So differentiated cells don't do mitosis. Differentiated cells are where you have specific functions. Like a skin cell is crammed in there and creates a watertight barrier, right? So this differentiated cell does the specific functions you need it to. If you want the cells in the pancreas that create your insulin, you have to go through the differentiation pathway to get those cells. All right, liver cells, differentiated. So normal cells undergo differentiation. So fully differentiated cells no longer divide, that sort of thing. Okay, so we've talked about how we get through the cell cycle, how we start it and stop mitosis. Now let's talk about just completely ending the cell cycle. So remembering that the cell cycle is the life cycle of a cell. Normally, that goes interphase to mitosis and you start over. All right, so you have your interphase. And then you have your mitosis. And then you have a new cell and the whole thing starts over. It's a cycle. So how do we end the cycle? How do we just stop the cycle? The process is called apoptosis. This is cell death. You can end up with apoptosis from two sources. In one source, you have programmed cell death. The cell ends its own cycle. In other words, you have a limited number of divisions. Wow. Or a limited amount of time. My digitizer is going nuts. Limited time. And then you have a gene that triggers the cell's own death. So, programmed cell death. You have a limited number of divisions or a limited time of being alive, and then a gene triggers the cell's own death. That's programmed cell death. You can also have apoptosis signals. For instance, there is a type of immune cell produced by your body called an NK cell. NK stands for natural killer cell. The natural killer cell, so an NK cell, well, NK stands for natural killer. The NK cell, the natural killer cell, releases apoptosis signals to cause your body's cells to kill themselves. So this releases signals and kills cells. It is worth noting that this is useful. Having a cell that's whole job is to kill your own cells. If you have a viral infection, viruses replicate inside your cell before rupturing out of the cell and going off to infect more cells. If an NK cell can detect a cell that is infected, it can cause that cell to kill itself before new viruses are replicated and sent out. So it can cause an infected cell to kill itself. So that's why you have natural killer cells. They're there to defend you by nuking bad cells or infected cells, right? So it's that dude in the zombie movie that shoots the person who got bit. So programmed cell death or signal cell death from natural killers. Natural killers will become important in the cancer lecture later. Okay, now that we've talked about how the cell pathways are regulated, like a stem cell has to be turned on to undergo mitosis and then turned off to stop undergoing mitosis. And then the daughter cells undergo mitosis, and then you get, and that's the differentiation pathway. So, main concerns. Mitosis has to be turned on and off. And when you turn on mitosis, you go through a differentiation pathway to get cells that just don't do mitosis. So now we're set up to talk about cancer. So oncology is the study of cancer. Onc or onco is referencing cancer. So, study of cancer. $20 to 25% of American citizens, end up with cancer. So, 20 to 25% of people in the U.S. end up with cancer of some sort. So this is considered your cumulative lifetime risk of getting cancer. That means, like, it's not a quarter, you're not like flipping quarters to see if you get cancer. It means that the longer you have been alive, the greater your chance gets, so that by the time you hit the end of people's lifespans, 20 to 25% of people have ended up with cancer. So the cumulative lifetime risk is different from an acute risk. An acute risk is like, if I jump this, I have a 50% chance of landing on the other side of this rail or something. Like if you're skateboarding and you jump onto a rail, you have like a 50% chance of making it. That's an acute risk, an instance of risk. A cumulative lifetime risk is the risk that builds over a lifetime. So that's important to know. So now that we know that 20 to 25% of the population is going to end up with cancer, let's talk about what cancer actually is. So cancer refers specifically to cancer cells. So cancer cells are cells that are replicating, right? So cancer cells replicate. So first off, cancer cells were all originally your normal body cells. You don't like get cancer from an outside source. Something goes wrong with a body cell in order to get cancer. So most often cancer is caused by mutations, right? So a mutation can turn a normal body cell into a cancer cell, right? So you can have sources of mutation can include like chemical sources, mutagens, So outside chemical sources. But they can also include replication failures and repair failures. So basically, anytime a cell is damaged, damaged cells need repair, or really damaged organs. Let's stick to the organ level. Damaged organs and tissues need repair. And to repair, you need more cells. And so, to get more cells, you trigger mitosis. And remember, in the S phase, you duplicate all of the DNA. Well, that has just a random chance of error all throughout it. Every time DNA is duplicated, you have a chance for mutation. And mutation has a chance of a cancerous mutation. So, cells replicate just normally over your lifespan. Red blood cells have something like a 6-week lifespan before they start to break down. And so you're constantly replicating new red blood cells. And any time that you're replicating new red blood cells in the hemopythetic pathway, you can have a mutation that is in the wrong spot and results in a cancerous mutation. Generally not of red blood cells, but of white blood cells. So that'd be like lymphoma. That's just a type of cancer that can occur just sort of randomly, because you're constantly making new cells, because cells have a life cycle and they break down over time and have to be recycled and replaced. And then anytime you have damage, like say you're breathing in something that's like maybe a plant material wrapped in paper, and you light it on fire and then you breathe in the result, that smoke irritates the respiratory pathway. That irritation is actually the smoke causing some damage along the respiratory pathway. And anytime you get damage to organs and tissues, you need to replace cells. And anytime that happens, you can have things go wrong. So the more often you're getting damage, the greater your chances of getting cancer. So originally, cancer cells were normal functioning human cells. But something caused a mutation in the DNA, whether it's just random chance from normal processes or exposure to outside chemicals, a mutation occurred, and two things happen. One, it starts to undergo infinite mitosis. So this is kind of scary. For one, this can happen in differentiated cells. If you produce a white blood cell with this mutation, that white blood cell, normally the end product of differentiation and can't mitotically replicate, manages to start undergoing infinite mitosis, and it disregards all stop signals. Right? So disregards stop signals, undergoes infinite mitosis, and it does that without start signals. So, not great, because as these cells undergoing infinite mitosis crowd up an area, they build a tumor, a neoplasm, which is a new growth. So, tumors are masses or growths of cells. Right? Neoplasia is probably the best tense to use. Let's not get into the grammar of it, since I'm only going to require you to know neoplasia. An important thing to note about tumors is that they are non-functional. This is because cancerous cells tend to lose their differentiation. I'm going to put lose in quotes because it's faster than become undifferentiated. So cancer cells in these mutations that allow them to start mitosis without start signals and just undergo mitosis infinitely while ignoring stop signals, they tend to be undifferentiated. So tumors are non-functional. Cancerous tumors. Non-functional tissue taking up space. This is, by the way, why they tend to reduce organ function. Because if you have your liver here and you get a cancerous tumor growing, as it grows, it displaces normal cells and just takes up a hunk of tumor while not doing tumor things. So you have reduced tumor function by the amount of space tumors take up. So organ failure due to cancer is because tumors are non-functional. Now, this is all cancerous tumors. There are non-cancerous tumors. These are called benign tumors. So a benign tumor is the result of cells, and it will grow over time. So you have these mutated cells, and these mutated cells start undergoing mitosis. But interesting things about benign tumors, right? So one, benign tumors tend to have a bit of tissue surrounding them. Called the capsule. So that's the tumor capsule. So you've got this tissue surrounding the tumor growth. So the tumor it's not like rigid, so the tumor can still grow, But a big thing about the tumor capsule is that cells from the tumor do not spread. The big thing about cancer is whether or not it started to get to different parts of the body. So benign tumors are a continually growing tumor, but it has this capsule and the cells do not spread to new areas. Benign tumors are bad, right? Generally, any growth crowding an area is bad. Benign tumors can be functional cells. In other words, they can be differentiated. I had a ferret named Loki who had insulinoma. So if you see OMA, you're likely referring to a type of neoplasia. My ferret Loki's insulinoma was benign tumors. So he didn't have cancer, but he had benign tumors in his pancreas, and they were tumors in the cells that make insulin. And that meant that all these expanding tumors were cranking out insulin. And so Loki had massive amounts of insulin in his body. I had to give him steroid shots to basically shut down insulin production because or drastically reduce insulin production. If it shut it down, he'd die. But the steroids I gave him drastically reduced insulin production because what ultimately did him in was when the steroids lost effectiveness, because they lose effectiveness over time, and he produced too much insulin, and it basically robbed all of his blood of glucose, and it robbed his brain of glucose. So his brain didn't make enough ATP, and he had a big seizure, and he died, which is a sad story to mention how those were differentiated cells. Those were cells that were actively making insulin. So benign tumors can be functional. They can be differentiated. This is to contrast with cancerous tumors, malignant tumors. Okay, so a big thing about malignant tumors, cancerous tumors, is that they have no capsule. That means that they can be very dangerous because without a capsule, right? So here's my tumor growing. And here is a blood vessel. Without a capsule, cancer cells can enter the bloodstream, and that allows them to spread. They allow cells to spread to new parts of the body. Oh, that's dangerous. Spreading to new parts of the body has its own specific term, right? Who knows the term for when your cancer has spread? Starts with an M. There's 7 of y'all in here. Maybe one of you knows. Take a sip of this. No? Gee. Metastasis is the spreading of a cancer to a new part of a body. So metastasis is the spreading of a cancer cell to a new part of the body. So when you have a metastatic tumor, that means that tumor started in one place, right? My mom's friend who died of colorectal cancer died with colon cells creating tumors in the wall of her heart, in her liver. I don't think they reached her brain. I think they did reach her lungs. So if you pulled those cells, if you pulled a sample of those cells, you would find that they were colon cells. So that's how cancer spreads and kills you. It enters blood vessels or lymph vessels. Your lymphatic system is like a series of vessels that run alongside your veins. So here's like a little lymphatic vessel as well. Basically, the lymphatic system has a bunch of little nodes all throughout your body, and they pick up water. So when your blood supplies oxygen to cells, it's actually little capillaries, the smallest blood vessels, and oxygen, water from the blood plasma diffuses out of capillaries and carries oxygen to those cells, right? And so that means water has left the blood. To get the water back into the blood, lymph nodes pick up that excess water and it travels through lymph vessels until it gets to veins near your heart and dumps the water back in. So, yeah. That's lymphatic vessels. That can also be a method for cancer cells to move. Ultimately, they end up in your blood. But so yeah, metastasis, not ideal. So here's a benign tumor, and here's cancer with no capsule, thus allowing the cells to spread. So cancer cells invade and infiltrate an organ. So for instance, if you've got a liver, which I'm willing to guess most of you do, and you get a cancerous mutation in some liver cells, those cancer cells produce a neoplasia, a growth, and it invades the liver cells, and anywhere where that growth is cells that used to do liver stuff and now don't do liver stuff. So as they invade and infiltrate, they reduce organ function. and they just continuously undergo mitosis, continuously replicate, disregarding all stop signals. And they are undifferentiated, like I said, so they aren't doing liver functions. And they replicate infinitely, so that the tumor just keeps getting larger and larger. So not great. A liver, by the way, has a ton of blood vessels in it. So there's my ton of blood vessels. And so if I get cancerous cells breaking into the blood vessel network, it can move through. It'll spread to different locations in the liver. Right? As it spreads along those vessels, it spreads to new locations and gives you more tumors in the liver. And eventually it can spread into the vessels that go to different spots. So if you have local cancer, that means it's only spread to nearby organs. So, you know, you might get liver cancer spreading to like your stomach or something because it's right up against it. Anyway, not great. So another scary thing about cancer cells, right? So let's get a newly growing tumor going on. And let's put a blood vessel nearby. So here's a blood capillary carrying oxygen and nutrients. And they diffuse out of the capillary into the surrounding tissue. And diffusion is pretty useful as long as you've got a lot of blood vessels. But one of the problems facing a cancerous tumor is that the larger the tumor grows, the less capable these cells are of getting nutrients. Right? Larger tumors, tumors themselves have a very low surface area because they're basically just a growth, right? Folds give you surface area. I think I've talked about this before, but if you imagine these were pieces of string and you cut this one and you cut this one and you pull them apart, the amount of surface area on the round shape is way lower than the surface area of the folded shape. So, tumor, having basically a round shape, has very low surface area. So, basically, diffusion has, you know, diminishing returns. It can only get so far. So when we're just getting oxygen and nutrients diffusing in, that's like our diminished returns point. These cells are O2 and nutrient starved. Diffusion just doesn't cut it to get oxygen and nutrients down to those cells. That's because the tumor grew too large for it to be effective. But tumors, cancerous cells, have a surprising ability. It's present in all cells because all cells have all the genes, but cancerous cells activate this, and they secrete angiogenesis factors. So angiogenesis factors create new blood vessels. In other words, angiogenesis factors stimulate new blood vessel growth into the tumor, and they can redirect existing vessels into the tumor. So I had this blood vessel that moves past here, and I stimulate some new vessel growth. Into the tumor. And it can also redirect the blood vessel into there. Right? So, this ain't great. This is not great, right? All this tissue where it was at is starving. Wow, digitizer, having issues. Ugh. The tissue gets starved when the blood vessel is redirect, and the new blood vessel also helps feed it. And so we solve the problem of diminished returns on diffusion. We solve our diffusion problem. The tumor gets to live. Hooray! Sucks. Okay, so it releases angiogenesis factors that cause blood vessels to create new growth into the tumor and redirect blood vessels into the tumor. So, stimulates new growth that supplies oxygen and nutrients and diverts existing growth, redirects existing growth, and starves the original tissue that those blood vessels served. So now it's not just causing organ damage because the tissue it replaces is functional, killing functional organ cells by pulling their blood supply. And it is increasing its ability to metastasize and move cells to a new place because it has pulled blood vessels right into the tumor. So, because we have angiogenesis and redirection, new blood cell growth and redirection of blood growth, blood vessels, new blood vessel growth and redirection of blood vessels into the tumor, it creates a really convenient highway for cancer cells to get into the bloodstream, and they'll create new tumors. They travel and anywhere they end up, they make new tumors. So they travel through the blood, they travel into the capillaries, and they create new tumors all over the place. So at first, it's just new tumors in the organ itself, but the longer the tumors grow, the greater the chances of the cancer cells getting further and further into the bloodstream. So you can have local cancer in which they haven't metastasized too far away. They're either contained in one organ or that organ and the organs surrounding it. And you can have distributed cancer where it's metastasized to distant parts of the body, creating new tumors. Not great. So here's a little cancer timeline, right? So we have a mutation, and it starts to divide infinitely. And so you start to get these cells infiltrating the tissue. And then you have another mutation. This is a sample pathway. Another mutation allows it to start growing the tumor. And my tumor grows. And in this example, you have another mutation that allows the tumor to invade the tissue. And then that tumor cell invading the tissue ends up getting into lymphatic vessels or blood vessels. And you get distant tumors. Tumors spreading to new parts of the body. So let's go through some types of cancer cells. So, carcinoma. A carcinoma is a cancer of the muscle. Wait, am I remembering that correctly? Let me just confirm that. Every once in a while, I go through my lectures and I just go, wait, does that sound right? Epithelial cells, it's skin cancer. Sarcoma is muscle. Jeez, brain. So skin cancer is a carcinoma. Sarcoma is muscle cells. It's also bone cells, and it's other connective tissue. So this is like linings around organs. Linings around body cavities, tissue that helps hold skin and muscle together. So that sounds an awful lot like etc. So muscle cells, bone cells, other connective tissue, those are sarcomas. Leukemia is cancer in white blood cells. White blood cells are leukocytes, hence leukemia. Then we have lymphoma, which is cancer of the lymph nodes. Which is my bad because I earlier said white blood cells cancer is lymphoma. So that's on me. So lymphoma is cancer of the lymph node cells. So carcinoma, sarcoma, leukemia, lymphoma, these are some good examples. I don't know in what situation you might have to, come up with examples of cancer cells. Who knows? It's a mystery. Let's see where we are. Slide 14. I think we could get through some basic cancer causes. So let's talk about what increases your lifetime risk of cancer. So there are predisposing factors. These are generally genetic issues. So predisposing factors are genetic factors, hereditary factors. Then there are carcinogens. So these are things that you can be exposed to. Often we might put these under the heading of lifestyle factors. Then there's viral. So some viruses actually induce cells into becoming cancer. So that's a crappy way to get it. So first let's talk about predisposing factors, factors that are out of your control because you inherited them. So abnormal genes. There are genes that make it more likely, so genes that increase the chances of specific cancerous mutations. So they increase the chances. These are not genes that give you cancer. These are genes that make it more likely that at some point in your life cycle, something will go wrong and you'll get a cancerous mutation. So this would be through replication and repair. So abnormal genes increase the chance of cancerous tumors during replication and repair. So for instance, there's the BRCA1 and BRCA2 genes. The BRCA1 and 2 genes. Breast cancer genes. These are oncogenes. Luckily, these are recessive. So if one parent has them and the other parent doesn't, if you get, but your other parent does not have them, then you're going to get a dominant from one parent and a recessive from another, and the breast cancer genes won't be expressed. So, hooray! This is where I just say that BRCA1 and 2 increased the chance of getting cancerous mutations in the cells in the breast that produce that specific type of breast cancer. By the way, it is worth noting that there are many many mutations that can cause different cancers. Heck, there's different kinds of cancers of the same organ, right? You could have 2 different types of breast cancer. So it just depends on what mutation you have. Is dangerous. Got about 15 minutes left. So, oncogenes are dangerous. Mutant breast cancer 1 and 2 genes. Lifestyle predisposing factors. So these are things that you do during the course of your life that increase your lifetime risk of replication and repair problems. So, diet can increase your lifetime risk of cancer. For instance, obesity increases your risk of reproductive system cancer. Obesity is often a catch-all term in medical science based on the old BMI. As it turns out, obesity is sort of like a highly specific thing to each person. Each person has healthy weight ranges for themselves. And so when we say obesity increases your risk of reproductive system, that would be whatever obesity is to your body system. Basically, obesity is when your blood tests start indicating that you have problems associated with obesity. So that's why blood tests are important. Sometimes, you know, your weight is not all humans have the identical body shapes. We all have our own unique gene variations, and that can result in variations in body frame and natural body weight. So, like, doctors will shorthand use the BMI chart and say, well, you're obese. But, like, for instance, people who are tall, my brother is 6'6", and a normal weight for him often pings like obese, because, you know, the taller you are, the more weight you have. It's especially a problem in females. The obesity charts for females are, the BMI charts for females are different from the BMI charts for males. And so, like, a really tall female would have a BMI chart saying she's obese, whereas if she lost weight, she'd be unhealthy because, like, you know, anyway. Obesity is highly specific to each person, and you'd know if you were obese because your blood tests would start showing you're obese. So anyway, obesity increases reproductive system cancer risk. Nitrites increase your risk of colon cancer. Let me write that down. Increase risk of colon cancer. So nitrites you find in cured meats. So they often have added nitrates and nitrites. And eating nitrates and nitrite-rich products increases your lifetime risk of colon cancer. Now, it is worth noting that nitrates and nitrites are actually a really important food additive. If you start buying uncured meats, you will notice in big words they say, keep it X temperature, use in 1 week. that often isn't present on cured meat packages. This is because nitrates and nitrites prevent Clostridium botulinum from becoming an active bacteria. Clostridium botulinum secretes botulism toxin. Botulism toxin is deadly. So even in relatively small concentrations, there are people like what it does is it causes muscle cells to be unable to contract. So a muscle cell relaxes and then cannot contract. So this is my muscles here contracting to pull on these tendons to get my fingers to move into a fist. If I had botulism toxin being produced around here, I'd relax that muscle, and then that'd be the end of that, right? So botulism dangerous, especially once it hits the diaphragm that controls your lungs and you breathe out for the very last time. So nitrates and nitrites prevent botulism bacteria, which are just soil bacteria and present quite frequently on meat products because most of our meat animals live in dirt. So they're present on most meat things, and so they add nitrates and nitrites to meat products to prevent botulinum from germinating and producing active bacteria. So if you eat food that's low in nitrites, like I do, I often eat uncured meats. You need to stick to those recommendations on temperature and time, because if you keep it at the wrong temperature or you let it go too long, you increase the chances that Clostridium botulinum will start replicating in there. The blackened, burnt stuff you get when you cook meat, that can have a carcinogenic effect. So it'll be similar to nitrites. To really make that a lifetime risk, it would require a significant amount of crunchy burnt meat, but worth mentioning. Then there's good old heavy drinking, right? Like alcoholism level of drinking. So this causes a lot of problems. Heavy drinking can result in cancer basically along the entire digestive pathway, like oral cancer. Whoa, my thing has stopped working. My digitizer. So, whoa, okay. PowerPoint force restarted. That's interesting. Well, it's 11:55. So rather than reopen everything and kill time that way, let's call it good. And I'll pick up with the type of cancers alcoholism causes. So I know, cancer lecture, not fun, but it should be done by Wednesday. So at least there's that. Yeah, I guess that's it for today. Let me jump. This is important because oftentimes when newspapers or other articles present your risks of cancer from doing certain things in your lifestyle, they are presenting relative risk without identifying that it's relative risk, right? So for instance, a high red meat diet For unprocessed meat, like if you eat a bunch of steaks or something, it is an increase of 13%. For a processed diet, like if you're chowing them all beef hot dogs, I guess, it's an increase of 20%. And these sound terrifying, but these are also relative risks. So cancer is a 20 to 25% cumulative lifetime risk. So when you adjust with the relative risk increase for unprocessed meat, what that comes out to is a 23 to 28% risk versus a 20 to 25% risk, and a 24 to 30% risk versus a 20 to 25% risk. So the absolute risk increase is not nearly as headline grabbing as the relative risk increase. So when you're reading stories about things, about dietary things that boost your chance of getting cancer, remember that these are always, almost always relative risks. Like if you chase down the citation, it's relative risk. And another problem with it, I wouldn't do that, but like certain preservatives and high doses are correlated with increased cancer chances. Again, these are all correlations, all the diet studies. We don't have a ton of causative pathways, right? So we have hypothetical causative pathways for nitrites. So we're getting closer to figuring out what that might be. We have definitely causative pathways for alcohol. And then that's where I talk about relative risk. And then here's a little thing on the study. This is all asterisk, which means this is not something you'd need to know for a test. So study limitations abound in studies on lifestyle and increased cancer risk, because how those studies work is they just send out surveys to like 10,000 people that say, please complete and return. And, you know, they just have to hope that people complete and return the survey. But they are, you know, essentially cold calling people. Like you just get a piece of mail and you're like, what is this? And it's got a survey, and most people would probably trash it, which is why response rates on these studies are generally very low. So they've got low populations. And then in order to make the studies more reflective, they even have to throw out more things, so they make sure that they're not overrepresenting one group versus another. So so they often have relatively loose controls. What that means is people self-report all this stuff, right? And I know when I go to the doctor and they ask how much exercise do I do on average, I really don't like checking the box that says like 2 hours a month or something. So I tend to overestimate how much exercise I do on habit. It's more than 2 hours a month, but anyway. Also, when people have bad habits, like smoking or drinking, it's your instinct, because those are considered bad habits, to underreport them. So people will fudge these things to a certain degree, and scientists have to try and take that into account as well. So there's some distinct study limitations on these survey studies, which make it so that if you read the citation and it turns out to be a survey thing, might not. You'd have to really examine their methodology to get an idea of how trustworthy it actually is. It may not be a good idea to just take every survey study on its word. For instance, way back when cigarette companies were doing advertising, the advertising agents would mail cartons of cigarettes out to doctors, just hundreds of doctors. And any doctors that wrote a thank you note would then get another thing mailed out to them, which was a survey about whether or not this is good for your health or harmful to your health. So only the doctors who sent a thank you note for cigarettes would get the, is this okay for you? And then they'd be like, 4 out of 5 doctors agree cigarettes are good for something or other, or whatever. It was the 50s, so cigarettes were considered healthy. Like, do you want to lose weight? Smoke cigarettes. It's great for you. Anyway, which, I don't know, dovetails right into the fact that the media often sensationalizes these things. For instance, relative risk versus absolute risk. It makes a way better headline to say that eating processed red meat boosts your chance of getting cancer by 20%, rather than saying boosts your chance of cancer by 4 to 5%. One of those is going to get more clicks. So, and that's a problem because often the people on science reporting beats are not scientists. Often with very little education in the sciences. So it's good to take a lot of these things with a grain of salt. Maybe read the article as to get an idea of the methodology. So anyway, back to stuff that is tested. Speaking of smoking, smoking is bad. I don't know if you knew this, but smoking isn't good for your health. I know, this is shocking information. So, like, definitely amazing. The stats are actually pretty terrifying. Smoking represents 30% of all cancer deaths. Right? When you look at cancer deaths, 30% of all cancer deaths can be attributed to smoking, which is surprising. Less surprising, 90% of all lung cancer deaths are attributed to smoking. Smoking is not good for you. Not only are there carcinogens in the cigarette smoke, just the very smoke that you're breathing in, the product of combustion, like the literal smoke, is an irritant for your lungs. So not only are you exposing yourself to carcinogens that can cause mutations, you're also exposing yourself to irritants, and an irritant is named so because it damages tissue, and damaging tissue requires repairs, and every repair has a certain rare percentage chance of kicking off a mutation, which has a percentage chance of being a cancerous mutation. So exposing yourself to not only carcinogens, but also smoke is a pretty rough way to treat your body. Smoking represents a 15 to 30 times increase chance of getting cancer. So your relative risk goes up by 15 to 30 times. So that's like it's pretty high, is what that is. I'm not gonna lie to you. That's so. The good news is that this is cumulative. So it's not like smoking a cigarette and now you're going to get lung cancer. Like, I guess that's possible if you're real unlucky when it comes to irritant repair. But the sooner you quit, the lower that increased relative risk is, because it's a cumulative lifetime risk. So if you spend less of your cumulative lifetime smoking, your increased risk from smoking is lower. I know, rocket surgery here, but sometimes it's worth saying. The sooner you quit, the lower that risk is. Like, you can manage the risk. If you've been smoking, it's not like you're guaranteed to get cancer. You can take steps that will head that off. 7,300 non-smokers die from cigarette smoke. So cigarette smoke, so cigarette secondhand smoke, and those deaths are largely cancer. So, you know, when you're walking on campus, eventually, again, would we one day walk on Auraria campus, maybe avoid the pack of people just puffing away on their cigarettes. Generally, there's just an increased risk of a bunch of cancer. Now, it's not just lung cancer, by the way. It increases your risk of oral and esophageal cancer. And, of course, lung cancer. So, not great for you. Just gonna throw that out there. It's my hot take, is that smoking isn't good for you. So this is a lung cancer tumor that has obstructed one of the tubes in the airways. So this is a healthy human lung with little to no accumulation of like dirt and other irritants in it. This is a tobacco smoker's lung, and what you see here is the tar that you get from the additional stuff that gets combusted. So you smoke the cigarette and you get the smoke in, and carried in the smoke is the stuff that you burned. And there's a lot of stuff burned in a cigarette. It creates tar in there. So this is a pretty common result of a significant amount of your lifetime spent smoking is cancer, right? So cancerous tumors infiltrate and invade the lung. And whenever they infiltrate and invade, the lung is basically packed entirely with respiratory stuff, like the tubes and the small little bowls. And so every tumor decreases your ability to breathe. And this is late stage lung cancer from someone who died of it. And you can see significant amounts of that lung are just non-functional masses, just tumors. So that person probably had like a 15% respiratory functioning at the time of their death. So lung cancer kills you by suffocating you, which is terrifying. Continuing along, stress is a predisposing factor. Who doesn't love to hear that stress is a predisposing factor? Because we all experience it. Now, the good news is that we're not talking about I have a test tomorrow stress, or I have a job interview tomorrow stress. We're talking about extended stress, like people who work an extremely stressful job, people exposed to trauma. They have rough immune systems. So the immune system increases cortisol secretion, which is a hormone that has a big cascading effect throughout the body. So the longer time you have elevated cortisol, the more drastic the effect. One of the effects is a suppressed immune function. Not completely, of course, but so let's put suppresses or depresses. There we go. Depresses immune system. So you have a decreased immune response. And this is highly correlated with your increased risks of cancer. So things you can do, sleep more, right? If you're constantly getting 2 to 3 hours of sleep a night, that puts your body under a lot of stress. Sleep is a regenerative process. Like your brain undergoes maintenance during sleep. Your muscles undergo maintenance. There's a lot of body maintenance that occurs during sleep. So try to get 6 to 9 hours, whatever seems best for you. But try to land in that area. Because when you're getting constant low amounts of sleep, you're getting elevated cortisol. If you have chronic sickness, if you're dealing with an illness that has a sort of constant effect or even just a chronic effect, that can lead to a stress response. So doesn't guarantee you're going to have a stress response. Some people sort of make peace with a chronic illness, but oftentimes you're going to have a stress response. If you get regular injuries, you're going to be in a chronic stress response. So these are not good things, and they elevate your risk of cancer. Just sort of any cancer, they elevate your risk of. We'll talk about this a little bit in the lecture, but your immune system actually has some heavy-duty anti-cancer effects. So when you depress your immune system response, that's one of the main reasons why extended stress leads to an increased risk of cancer, because your immune system has a decreased ability to handle cancer. So extended exposure to physical stress or mental stress, like trauma. So suppresses immune system. There we go. So that is the lifestyle predisposing factors. These are all things that you can do things about, right? You can't do things about the hereditary predisposing factors, genes that make it more likely that you get cancer. You can do things to manage the lifestyle predisposing factors. Now let's talk about chemical factors that increase your risk of cancer, right? So radiation, specifically ionizing radiation. So ionizing radiation are things that you think about when someone says radiation, like X-ray radiation, gamma wave radiation. These are all ionizing radiation. Not great. So there's a type of non-ionizing radiation. There's only one that I know of that increases your risk, and that is ultraviolet light. This is because ultraviolet light burns skin, and thus you can end up with a screwed up repair. So that's how non-ionizing radiation can increase your risk of cancer. So ionizing radiation like X-ray radiation, gamma wave radiation, I'm forgetting a few, but there's a bunch. those can increase your risk of cancer. By the way, radiation, when we say radiation, we're often talking about stuff coming from a source. The word radiation doesn't necessarily imply anything dangerous. This is why ionizing radiation and non-ionizing radiation are like good terms to use, because lots of stuff radiates out from a point, right? This is radiation. So visible light, that's radiation coming from a sun, right? So radio waves, those also radiate from a sun. So the term radiation in science is, you can shorthand it for ionizing radiation in the right context, but it doesn't automatically imply a certain dangerous type of radiation. So, for instance, microwave radiation is a lower power ionizing radiation. It is worth noting that when you look at the scale of places where you find types of radiation, we measure the type of radiation it is based on something called the wavelength. So energy travels in waves like that. Ionizing radiation tends to have a really short wavelength. Radio is over here. Radio is harmless. Even when you're exposed directly to extremely high power radio waves, the biggest danger you're going to have is like feeling hot. It can heat things if you have extreme high power radio wave exposure. So like if you went to your radio station's broadcast tower, climbed it, found the actual radio emitter, and then just sort of like hugged it for a while, you'd warm your body up potentially to dangerous levels. But if you stand a little bit further away, that goes away. Radio waves are not dangerous. Exposure to microwave radiation can be dangerous. So that has a heating effect. So it's worth noting, cell phones are in the radio spectrum. They're closer to microwaves, but they are in the radio spectrum, which means they do not exhibit any of the features of microwave radiation. They exhibit the features of radio waves. So people saying cell phones will cook your brain or cause cancer are likely saying that because some original person thought cell phones were so close to microwaves that they were dangerous. I mean, even if they were microwave radiation, it's unlikely that they generate enough power to be dangerous. So, but yeah, they are firmly radio and they are not dangerous. Just a little aside there. Back to radiation. Radiation is dangerous because it damages. Ionizing radiation causes a lot of damage. So when you're exposed to ionizing radiation, the energy flowing is absorbed by atoms in your body, and it causes atoms to kick off electrons. And so kicking off electrons changes the atom's charge. It can make it more reactive. The electrons being forced out of the atom can also like impact stuff and cause damage. That is a real shorthand for a much more complicated process, but the ionizing effect itself can cause damage. So being exposed to radiation causes radiation burns, and that's an effect of the ionizing effect of these radiation types. And anytime you get damage, you get an increased risk of cancer. And with the amount of damage radiation does, like when it hits your cells, it does large-scale damage to the genome. Like your DNA doesn't like take damage in one spot. It takes damage all over the strands. So then you have to repair the DNA. And when you have a whole lot of damage in a whole bunch of area, your risk spikes. So lots of ionizing radiation boosts your risk of cancer. But again, this is like all relative risk stuff. So, for instance, I love this one story about a Japanese guy who worked at the Mitsubishi plant in Hiroshima during World War II. Mitsubishi being a major manufacturer of engines for Japanese warplanes. And he was walking to work the morning they dropped the atomic bomb, and he happened to be late for work because he stopped and talked to a buddy. And so he wasn't in the city center when the bomb dropped, and so he wasn't vaporized. But when the bomb came down, the gamma, like a lot of the damage to biological stuff from a nuclear weapon isn't just the direct explosion. That's, you know, catastrophic within a certain range. But in a much larger range, you get exposure to a gamma wave burst. So the flash of a nuclear weapon is actually the gamma wave radiation being emitted. So this guy was walking to work. He wasn't vaporized by the actual explosion, but he got thoroughly saturated with gamma wave radiation. So that wasn't great. He took massive amounts of ionizing radiation damage in his body. So he did not end up with like severe radiation burns. And so when Mitsubishi headquarters needed someone to explain just what the heck happened to Hiroshima, they called him to Mitsubishi headquarters, where he tried to tell them it was a single bomb there. They didn't believe him, but Mitsubishi headquarters was in Nagasaki, which got the second atomic bomb. So he got exposed to a second gamma burst from an atomic bomb exploding. And he did not end up with cancer, and he had children, and their lineage does not appear to have an increased risk of cancer. So ionizing radiation is dangerous. The moral of that story isn't go sunbathe in nuclear test areas. The moral of that story is that whenever we have something that boosts your cancer risk, it's important to remember that that's not a guarantee of getting cancer. It is a boost. You do not want to expose yourself to a ton of ionizing radiation, but things like X-rays from, you know, doctor's office X-rays or CAT scans, they are exposing you to very, very minor amounts. So a lot of the ionizing radiation that Earth gets exposed to is like absorbed by the atmosphere. So if you take a flight in a plane that gets up to like 30,000 feet or so, you're getting more ionizing radiation than you would be at ground level. So I think like by the time someone in Colorado has lived, I think it's like 15 or 20 years in the state, they've gotten a cumulative dose of ionizing radiation roughly equivalent to the people outside Chernobyl. So, you know, just kind of interesting stuff. So exposure ain't great. The more you're exposed, the more cells take damage, and the more repairs have to be done, and the greater the chance of a screwed up repair. So The danger of ionizing radiation and ultraviolet light is that it causes damage, and it causes a lot of it. So, X-rays, gamma rays, which are the nuclear radiation. Radon is a type of ionizing. So radon is emitted by some radioactive elements that are very common in the soil in Colorado. So when you buy a house, one of the things they do is a radon test, where they go into the basement where there's the most soil around the house, and they test radon levels. And if you try and sell your house and you've got dangerous levels of radon, you have to put in a system that collects radon and exhausts it outside of the house. So that's a natural source of ionizing radiation. So non-ionizing ultraviolet increases your risk of skin cancer. So if you want to decrease that relative risk of skin cancer, maybe don't tan all the time. Like tanning is a great way to boost your relative risk of cancer. I guess if you want to sunbathe, but maybe don't go for the tanning beds. Tanning beds give you some concentrated ultraviolet radiation in order to induce tanning quickly. So you're generally getting exposed to more UV light in a tanning bed than you would get in laying around in the sun. So, or at least a more concentrated dose. The sun produces a lot of UV. Anyway, use sunscreen. That's a good idea. Okay, so those are some natural radiation sources that are carcinogens. Let's talk about chemical carcinogens. Tobacco itself, the tobacco used in cigarettes contains carcinogens, right? So the tobacco, so this is really tobacco smoke and not like the leaves from the plants. So if you smoke just tobacco leaves, you're not getting quite as much. Cigarettes get a lot of additives for things like flavor and smoothness, the characteristics of the cigarette smoke itself. They add a lot of stuff to the tobacco in a cigarette to give it like a more marketable feel to it. So that's why people have like a favorite brand, not just because of some random decision that they like this brand, but often because different brands have different additives, and that changes how the cigarette experience feels. But one of the side effects of having all those additives in there is that there's roughly 60 to 70 mutagenic carcinogens in there. So mutagens aren't just irritants. They don't just act by causing damage. Mutagens actually cause mutation. So it's not that they boost damage to the cells, and every time you repair damage, there's a risk of repairing it wrong. Mutagens just straight out cause mutations. So that's not great. There's a lot of pollutants that are dangerous carcinogens. So, for instance, pollutants that can be inhaled or skin contact pollutants. Right, so there's a variety of metals that are dangerous. For instance, good old asbestos is an example of a metal that, when inhaled, drastically increases your risk of cancer, your relative risk of cancer. The reason asbestos is so dangerous is that it is a extreme irritant. So asbestos is a metal that occurs naturally. and it has a fibrous structure to it. So it has like this fibrous structure of spikes of mineral. And these spikes break apart very easily. So you get like these extremely small asbestos particles that you breathe them in, and they kind of like shred things as you breathe them in. It's not like you're going to feel your respiratory system get shredded, but it is dangerous. So asbestos is bad. If you want to know why asbestos is everywhere, it's because, as it turns out, asbestos is not flammable. Which means it's an excellent fire retardant. If you take a building and you line it with asbestos and you set the building on fire, it will burn much more slowly and it will be more resistant to the fire damage. So asbestos is a fantastic fire retardant. It is an amazing fire retardant. It's just also extremely cancerous, right? Well, I don't know, but it's extremely carcinogenic. That's the word I wanted. So when people first discovered this property of asbestos, they put it in everything. Like couches had asbestos linings in them, pillows had asbestos lining in them, like buildings had asbestos linings. It was everywhere because it's just so good at suppressing flammability, right? You're not going to have yourself falling asleep after taking a few drafts of your healthy cigarette in 1930 and drop your cigarette on the carpet and have the whole place go up if your carpet is infused with high-tech asbestos technology. We didn't know asbestos was carcinogenic in the 30s when we put it in everything. So, but we do know it's highly carcinogenic now. But yeah, fun thing, by the way, if you want to say something is not flammable, I used to say that it is inflammable. Anybody do that? Say inflammable when you mean it's not flammable? Am I the only one? Inflammable comes from the word inflame. And inflame is to cause flame, which means inflammable means flammable. So, you know, you have to say not flammable if you want to say not flammable, unfortunately. So, I used to say inflammable all the time because in my brain it made sense to say inflammable. So, I don't know why, but there are a bunch of organic pollutants that can be dangerous if you inhale them or get skin contact. So, things like industrial or agricultural stuff, like generally chemicals called benzenes, or chemicals based on benzenes, are very dangerous. You have to follow the safety instructions very closely. So always follow safety instructions. generally speaking, when it comes to handling dangerous chemicals, if you follow the safety instructions perfectly, they're not going to be dangerous. So, you know, don't take a bath in your extreme, like, phosphine pesticides. So, yeah. Use protective gear. And then lastly, there's irritants. A lot of pollutants are irritants. So that means that they are inhaled pollutants. And they increase your risk of oral, throat, or lung cancer because they damage your airway. So 2.5 nanometer particles are generally considered pretty dangerous irritants. 10 nanometer particles are also considered irritants. Great news for all of us here in Colorado. You know what has a ton of 2.5 nanometer particles? Wildfire smoke. So, it's been a great summer for increasing relative risk of cancer, let me tell you. So, you know, there's always that. If you're trying to decide, like, whether or not to go outside, if it's rained recently, that usually knocks down pollutant levels an awful lot. But there's a website I've been using a lot, airnow.gov, I think it is. So it's pretty useful. It tells you, let's see, I don't know if my webcam thing is active when I'm doing this sort of stuff. So, but like, it just gives you a risk assessment there. So right now it's telling me that the air in Aurora has a moderate level of 2.5 nanometer particles. So if you have asthma or other respiratory illnesses, maybe don't go for a jog. So, you know, that's useful. Looks like someone started recording, so that's good. I completely forgot to start recording. So, good for all the people who are showing up. Bad for the people who didn't show up. Because if they say, hey, you missed something, I say, hey, it was during the synchronous class period. So, whoops, talk to your fellow students. So, a list of known carcinogens. Everything you eat, everything you breathe, everything else, except broccoli. On to viruses that cause cancer. So some types of viruses can induce cancerous mutations. So basically, the viral infection in a cell. So the virus enters a cell, and it induces a cancerous mutation. So that's not great. There's a limited number of viruses that can do this. Hepatitis B and C. Not ideal. EBV is Epstein. Is it 1P or 2Ps? Let me pop over to my spelling thing. 1P, 2 Rs. Epstein-Barr Virus. So hepatitis B and C, those increase your risk of liver cancer. right? Epstein-Barr virus increases your risk of lymphatic cancer. Come on. There you go. Which, if you recall, is lymphoma. Right? Then you have HPV, human papilloma virus. I think there's actually two P's there. Human papilloma, maybe there's not two P's. Now it immediately looks wrong. I hate human brains, people. I really hate human brains. Did you drool on my digitizer? You drooled on my digitizer. Human papilloma virus, or HPV. You might know this already because when we developed a vaccine, we talked a lot about how the vaccine, there's a ton of different HPV viruses out there, by the way. Generally, when it comes to viruses, there's multiple strains out there. And so the company that made the HPV vaccine, Gardasil, basically just analyzed which strains had the greatest risk of cancer and made a vaccine against that. It would be very difficult to try and make a vaccine against all the strains, but it does a pretty great job of protecting against human papilloma viruses, increased risk of cervical cancer. So, not ideal, these. The good news is that there is now a vaccine for Hep A, B, and in recent developments, C. So we now have vaccines for Hep A, B, and C. And we have a vaccine for HPV. The only thing we do not have a vaccine for in this list is EBV, Epstein-Barr Virus. No vaccine. Sad face with a tear. It looks like multiple eyes coming down, but whatever. So try not to get yourself Epstein-Barr virus. Actually, try not to get yourself any of these viruses, although human papilloma virus is insanely common in the population and generally has little to no actual symptoms. It's 2 L's, not 2 P's. I hate you, brain. So, HPV is extremely common in human populations, sort of no matter where you are in the world. HPV is extremely common, and the reason it's extremely common is that HPV is generally asymptomatic. You do not often find someone with genital warts. And the reason you don't often find someone with genital warts is because the immune system knocks out the wart part. You almost never express the actual genital warts. What happens is you just have the virus in your body in cells that are infected, but as long as your immune system is healthy, you don't express the genital warts. So if you were in high school and they showed you genital warts as some kind of scare tactic away from having sex, what they were showing you was people who were immune compromised. So immune compromised people have visible genital warts. So you can thank whatever health teacher you had. So Hep B increases risk of liver cancer, but there's a Hep A and B vaccine, and now there's a C vaccine. HPV increases your risk of cervical cancer. EBV increases your risk of lymphoma, and there's no vaccine. So I need to update this lecture to indicate that there's now a Hep C vaccine. So that was in the last 10 years and was a huge deal because Hep C is relatively common in like homeless populations. And till now there was no till the past 10 years there was no vaccine. So all right, natural defenses against cancer. That's right, we're gonna try and take an upswing here before we're gonna end high. So there are genes that code for an immune response that protects you against cancer. So these are tumor suppressor genes. Tumor suppressor genes are a natural defense against cancer. If you think about it, every time a repair occurs, there's a risk of cancer. So it honestly wouldn't make sense to have no natural defense against cancer. So you have coded into your genes defenses against cancer. Everyone has a large variety of tumor suppressor genes. Tumor suppressor genes activate during replication and during infection, and they repair malignant mutations. They repair and remove. Let's add that. So when the mutation goes wrong, your immune system can activate tumor suppressor genes and literally just yank out the mutation before the cancer starts to spread. So that's pretty cool. That's pretty great. So there are immune cell functions. I mentioned last time these cool cells called natural. They can induce cell death. So they can identify malignant cells and induce apoptosis. They secrete something which I think has a really cool name, TNF alpha. It doesn't sound amazing, but the actual words for TNF alpha are tumor necrosis factor. And it's a chemical that a natural killer secretes in order to literally cause a tumor cell to kill itself. So that's pretty cool. So they can remove and repair. So repress mutations, promote cell death. Your immune system actually recognizes a ton of cancers. So there are 10 million plus different types of cancerous mutations that the immune system can identify and nuke. So your immune system has a strong anti-cancer effect, right? It is built in. That means when we talk about cancer, we're often talking not about like all cancer, but just the cancers that your immune system can't destroy. And you might notice there's a lot less than 10 million. When we think of cancer, we're often thinking of cancers that evade or ignore or resist immune functions. So, so just a fun thing, which, by the way, means just in the act of, maturing into adulthood and growing and changing and whatnot, you've probably had more than a few cancerous mutations, just ones that your immune system nuked. So you've all had cancer already. So it prevents cancers from taking hold, prevents cells from becoming cancerous by destroying the mutations, and it kills cancerous cells by promoting this. So, it is pretty amazing in its ability to recognize cancer, and it can also recognize new types of cancer, right? So it's actually pretty cool just how few cancers there are that the immune system can't take care of. So your skin itself blocks carcinogens. That is because skin has melanin. Melanin blocks ultraviolet radiation. So melanin gives you resistance to cancer, resistance to damage caused by ultraviolet radiation. Let me get that thing out of your eye. It resists damage from ultraviolet radiation and helps reduce UV exposure, which is pretty cool. Skin is watertight, so it can block a lot of liquids from getting to the lower levels of your skin. So it can block viruses and pollutants. Like if you have to go through an area with a lot of asbestos, holding your breath goes a long way towards not getting cancer, which is pretty cool. Your body's mucus actually does a great job of handling carcinogens, right? So mucus lines your respiratory tract, so it can trap airborne particles. It can also trap pathogens. And you will, snot it out of your mouth. As far as the pathogens go, they're trapped and then they're nuked by the immune system. So you got a lot of natural defenses against cancer out there. So pretty great. Here's a little example of fun things you can do with immune cells and genetic engineering, which we will go into in a moment. So, surgical treatments for cancer are one of the classic treatments. Give me that. No. Drop it. There you go. Accidentally walking into a cord. So what you do literally is physically remove the tumor. So you just literally go in and cut out as much of the tumor as possible. So that's a classic. That was like the first treatment. Radiation is another early treatment of cancer because a concentrated dose will kill cells. So if you have cancer in a certain part of your body, whatever, here's a lymph node in your armpit, and it's producing a cancerous tumor. They may take a little radiation emitter and they will aim it directly at that tumor and give them enough of a dose to actually kill it. So radiation is a treatment which directly kills. cancer cells, really any cells, but honestly, killing cancer cells is a pretty good idea, even if it's going to cause some damage to the cells around it. Great example of radiation is the gamma knife. Gamma radiation is really shortwave radiation. It doesn't penetrate super far. And what happens with a gamma knife is that they have these two emitters, right? And they emit gamma radiation, but they emit it at a relatively low power. And this power is low enough to be harmless. But where the little emitters have a crossover, that's powerful enough to kill cells. So they call it a gamma knife because they can focus it around to different depths and really just try and pinpoint damage the tumor cells themselves. So it's actually pretty cool how that works, that you have this cool instrument that does pinpoint damage via radiation. So traditional radiation treatments back in the 50s just sort of nuked the whole part of your body where the cancer was and killed a bunch of stuff. But, you know, cancer bad. So killing cancer along with your own cells may suck, but it does kill the cancer. And lastly is chemotherapy. Now, chemotherapy is a, let's see, how do I want to put this? A targeted cocktail of essentially different drugs. Targeted is important because chemotherapy is specific to specific cancers. Using the word twice sucks, but I'm just driving it home. It's specific to specific cancers. They mix a chemotherapy cocktail for the cancer you have. So sometimes they do like intravenous treatments. I have a friend with a type of brain cancer where he just takes a chemotherapy pill. So chemotherapy does a lot to cancer cells, right? So chemotherapy can directly kill cancer cells. So they use chemicals that might target the receptors on cancerous cells, something that directly targets cancer cells. They can also do quite a lot to them. So they can inhibit replication. They can starve them of nutrients. That's just actual starvation. They can disrupt the cell membrane and break the cell apart. They can literally damage the DNA of the cell. If it does enough DNA damage on average, it kills the cell, regardless of whether or not it's cancerous. And they can inhibit repair of DNA damage. So, chemotherapy is highly targeted. It's tailored to the specific types of cancers. In general, chemotherapies kill fast replicating cells. So this does a lot of damage to fast replicating cells. Sort of a collateral damage to that is hair follicles are fast replicating. So the production of hair is done by quick replication of keratin-filled cells. So your hair is largely dead, but the living follicle replicates quickly in order to actually grow hair. Your hair grows really fast compared to other things in your body. Intestinal linings can take damage. Stomach linings can take damage. These are things that have to continually be produced to protect your digestive system and lubricate your digestive system. So often side effects of chemotherapy are losing your hair and also nausea. It can have effects on arousal responses too. So again, just generally make you feel terrible. But the thing about it is that it is killing cells. For instance, when it comes to childhood leukemia, we have an extremely well-developed chemotherapy cocktail, which has gives people with childhood leukemia an 80% survival rate. That is a pretty nutty 80% like survival rate. That's nuts. Hodgkin's lymphoma 1 and 2, like this is, if you catch it in stages 1 and 2, it has a 90 to 95% survival rate. So chemotherapy can be extremely effective. It makes you feel like garbage. Oftentimes people say like, oh, chemotherapy makes you sicker than the cancer. So you'll have people who bail on chemotherapy and they start to feel better. And they're like, oh man, chemotherapy was what was wrong with you. But there's this myth people like to think about called the beautiful death, right? Like, oh, I have accepted that cancer will kill me and I've gone off the chemotherapy and I will just live my life to the fullest until I pass from it. And what you're actually passing from are excruciatingly painful tumors throughout your body that can cause you to have trouble breathing, reduced cardiovascular effectiveness. When it hits your intestines and stomach, you're constantly vomiting. You're vomiting blood, you're pooping blood, you're diarrhea. Like, there's no beautiful death from cancer. Like brain cancer is like your best chance to go out without experiencing a ton of horrific pain, because later stage, like brain cancers can't really travel. The blood vessels in your brain are actually surrounded by a type of brain cell that basically creates a barrier. So brain cancers generally don't metastasize to places other than the brain. And when a tumor gets big enough, it just puts you to sleep forever. Your family has to sit there and watch you poop yourself and slowly waste away and die. But at least I guess you're asleep. So that's like the closest you can get to the whole I'm going off chemo and I'm going to live my life to the fullest. Cancer is a horrific death. Like, there's no beautiful death in cancer. So just throwing that out there. Common misconceptions. Like, you get movies where people are like, I'm just going to live the rest of my life. And then it's like a beautiful scene where they like hold hands and they pass away. And it's not at all that. So let's get a little happier than my rant about the beautiful death. Immunotherapy. So let's talk about different immunotherapies. One is P53 Gene Therapy. So an interesting thing about a gene. The scientists identified a specific gene called P53. And cancer cells don't have P53. So scientists were curious what happens if you give cancer cells P53. So what they did is they took a type of virus called an adenovirus, and they pulled out all of the virus genome. So they removed the pathogenic genome, and they added P53 to it, and basically they infected cancer with P53. So they just took a big dose of this adenovirus vector, and they injected it into a site of tumor cells, and just put a whole bunch of viruses in there, and then a whole bunch of tumor cells will get hit by it. And what happened was, when you add P53 to a cancer cell, it induces apoptosis, or apoptosis, whatever you want to say. So it's absent in normal cell, or it's absent in cancer cells. In normal cells, it actually has no effect, which is great, because that means you can just sort of flush it through an area with cancer, and it won't harm normal cells. It won't induce apoptosis in the normal cells that get exposed to your adenovirus vector, which is pretty cool. So P53 gene therapy is actually a pretty big potential source of cancer therapy. It's really expensive to prepare this adenovirus vector, though. It's not ideal. So it's really expensive, which is probably why it's not been granted common FDA approval, because it's really intensive to engineer enough viruses to have an effect. You have to deliver enough viruses in one area to get a concentrated effect. And that's hard to do, because when you remove the viral genome, it's not like you can take an engineered virus and then replicate it. You have to treat a whole bunch of viruses and then inject them. So P53 is amazingly effective, but at the same time, it is not ideal cost effectiveness wise and labor wise. Like, it's so time intensive. And if there's cancer cells that just through random chance don't get hit by that adenovirus vector, they're just going to start replicating again. But extremely promising. However, there is an FDA approved treatment. And it is an FDA approved treatment for leukemia. And it is a lentivirus genetic therapy treatment. And a lentivirus vector is a fancy way for talking about something that people might find terrifying. So, lentivirus is literally HIV. So what they do is they take HIV and they remove the pathogenic genes, and then they insert new genes. So does anybody happen to know what kind of cells HIV is extremely effective at infecting? Come on, someone. Participation time.
I'm honestly not sure.
HIV infects. Thank you for speaking up. That at least lets me continue. I'm going to force people to talk. HIV infects white blood cells. White blood cells are immune cells. So these are cells that could have an anti-cancer effect. So immune cells have their own anti-cancer properties. And the big problem with trying to use the immune system to attack cancer is that it just can't recognize certain cancers. So the whole idea of removing HIV's pathogenic genes and inserting new genes is that you can insert genes to recognize new cancers. So specifically, the type of cancer that person has. So you insert a gene to recognize. And so they did this. They took a bunch of white blood cells. I think it's actually lymphoma, not leukemia. Anyway, they took a bunch of white blood cells and they pulled them out, healthy white blood cells. They infected them with this engineered HIV and they put them back in the body, and the recognition gene actually caused these white blood cells to attack and kill the cancer cells. But there wasn't enough of them to do the job, to eliminate the cancer. So they did a second round of experimentation. They set up an entirely new experimental group, and they, let's see, skip that for a moment. So in the experimental group, what they did next is they added a gene to cause, how do I want to say this? Wild replication levels. So what happened was they then got white blood cells that could recognize a cancer and replicated themselves enough that there were enough white blood cells that recognized the cancer that they could wipe it out. So the vector inserted the gene. I have promising because this is old. This has been FDA approved. And I think I've already mentioned that it inserts the gene. This is just unnecessary there. I love this comic. This is from XKCD. So let me just hide the ink on the slide. Where's hide ink markup? But, okay, so I have blood cells growing out of control. So you're going to give me different blood cells that also grow out of control? Yes, but it's okay because we've treated this blood with HIV. Are you sure you're a doctor? Almost definitely. So this treatment is actually in use. It has been deployed, which is pretty cool. So there we go. This is just sort of a little summary of treatments we have. So you can have surgery to remove the tumor. You can have radiation and chemotherapy to kill the active cancer cells. You have P53 gene to induce this. Right. You have just regular immune responses. And then part of chemotherapy is these drugs here. So I'm going to circle these effects here. These are also chemotherapy effects. So, pretty cool. Lots of different cool treatments out there. So, some cancer danger signs. Unusual growths. So, unusual growths that perhaps weren't there recently are danger signs for cancer. Don't want those around. Unusual bleeding or discharge. Right? My friend's dad, who recently passed away of pancreatic cancer, discovered his pancreatic cancer because he started vomiting blood, and there was no clear reason for him to have done that. So unusual new growths, unusual bleeding or discharge, lumps lumps or thickened tissue. These are danger signs. Sores that won't heal. That's a pretty decent danger sign. So it's either like maybe a cold sore from a virus, or some cancers can cause sores. Bowel or urinary dysfunction. So for instance, colorectal cancer, pretty much you get tons and tons of diarrhea. So bowel or urinary dysfunction. So often we're talking about like sudden changes, things that aren't explainable by like getting a virus or an infection or a bacterial infection, right? You are otherwise seem healthy. Unexplained or unusual hoarseness and cough can be a danger sign of cancer of the respiratory tract. Moles and warts. So new moles and warts, especially pay attention to the size and the margin. So this here is an irregular margin. Common moles tend to have a pretty regular margin. They tend to have pretty circular margins. Whereas skin cancer growths will often have irregular margins. And they'll also change in size. They might get smaller, they might get larger, and there are observable changes. And then lastly, unusual or unexplained difficulty swallowing or digesting. These are all danger signs for cancer. So, you can manage your cancer risk to a certain degree. Like that base 20 to 25% cumulative lifetime risk of cancer is just sort of your lifetime risk from being alive. But 75% of the relative risk of cancer comes from predisposing lifestyle factors. So avoid and protect yourself from UV radiation. That's a big one. You can avoid skin cancer pretty effectively. Quit smoking. That is a huge benefit. The moment you quit smoking, you stop exposing yourself to carcinogens and irritants. The very moment you quit smoking. Early detection. So follow the doctor-recommended visits based on your age, So like at a certain age, they start wanting to do colonoscopies in men and breast exams in women. So follow the doctor-advised regimen on that one. It's changed since the 80s. Like women in the 80s, like over 30, they were like, come in every year to get your breasts smashed and a picture taken. And my mom is happy that that changed to like once every 5 years, but unhappy that, you know, she was over 30 or 40 in the 80s. And so that was not a good time. Stop aging is a great way. By the way, in addition to following the doctor advised stuff, if you have a danger sign, maybe go see a doctor. Like, go to a doctor if something irregular is occurring in your body. My friend's dad got to live an entire year with pancreatic cancer, which is pretty amazing because the very moment he vomited blood, he went to the doctor. Somebody might ignore that, feeling like, oh, maybe they're sick, or I'll wait to see if it happens again. And pancreatic cancer is a very rapidly growing cancer. So he got a whole year with his family to, you know, sort of say goodbye. So that's pretty amazing. Don't age anymore. That's a great one. So everybody just get on that. Because that cumulative lifetime risk of 20 to 25% is just the increased error rate as you've lived a long time and as you get older. So as you get older, you have increased errors, you've taken more damage, you have decreased immune responses. So there you go. That's your methods to stop your cumulative lifetime risk of cancer. Generally, the elderly go out, if they're healthy, from cancer or heart disease. One of those, or heart attacks. One of those two tends to be the most common cause of death in the elderly. Puppies, because this one has been depressing. So there we go. Let's see what time it is. 11